This is lecture 9. In lecture 9, what we're going to do is go through examples of eukaryotic microorganisms and some major features of them. Now, when I'm talking about eukaryotic microbes or microorganisms, I will discuss in this order fungi, and then algae, and then protozoa, and then helminths, which are worms. All right. Now this lecture, similar in some ways to the lecture on the part of the lecture on viruses where I went through different families, and it wasn't so much memorizing all the family names as it was knowing some important members of those families. Here it's essentially a survey of important members of eukaryotic microorganism groups that cause disease in humans. Okay, first of all, the fungi are, of course, eukaryotic. They are chemoheterotrophic organisms. They use chemical organic compounds to they break them down to get energy, and they also use them as a source of carbon to build up organic molecules. Just as we are chemoheterotrophic organisms, the fungi are also. Most, not all, fungi grow on things that have already died. They are aerobic or facultative anaerobic organisms. There's only about 200 of the 100,000 species of fungi that can be pathogenic in either humans or animals. As I said, they grow on dead things mostly. Most are decomposed things that are dead. Now, there are major categories. There are molds, which are multicellular fungal organisms. They have a filamentous structure below ground, which helps them to take up nutrients. They are also form an important web of these filaments form an important web for transferring nutrients between fungi and trees. Fungal organisms grow on the surface of roots underground. But we're not so interested in trees as we are in things that cause disease in humans, so let's move on. They're identified using biochemical tests or morphology or by the spore type. The yeasts are single-celled fungi, and their identification involves biochemical testing alone. And then there's a group that grow as mold or yeast. They are dimorphic fungi. They can grow either as a mold, multicellular phase, or a yeast, a single-cell phase. And in fact, most pathogenic species are dimorphic fungi. Mycology is the study of fungi, and it's a big topic, and I took an entire course just in mycology. Not an easy thing to swallow whole, particularly because of the similarities and subtle differences between different groups of fungal organisms. So we're going to go through the major characteristics here. When we look at molds, the multicellular fungal organisms, the main body consists of long filaments, so they are filamentous, as I said, and the main body, or thallus, the filaments are called hyphae. And when you look at a mass of hyphae, it's called a mycelium. All right, so the fungus that you see actually coming up for example, as a mushroom growing out of the ground, has a huge network of filaments underground that form its mycelium. Yeasts or unicellular organisms divide evenly or asymmetrically, depending upon whether they divide by fission or budding. And the dimorphic fungi are, again, most of the pathogenic types of fungi are dimorphic and are yeast-like at 37 degrees, but are mold-like at cooler temperatures, 25 degrees. Now, different groups of fungi have a life cycle, and typically there's a sexual cycle and an asexual cycle. This is one example. These are a type of fungus that has what's called an ascus. That's its key point. The differences between different types of fungi mostly reside in the structure that holds the spores. And there will be sometimes sexual produced spores, and other times it produces another structure for its reproduction by asexual means. And the differences between different groups of fungi are subtle, and they're easily confused, and that's not what we're going to teach in this course. So you don't have to worry about that. The main point being that they typically have a sexual part to their cycle where they have sexual reproduction, and they also have asexual reproductive part of the cycle. There are sexual spores and asexual spores, and as I said, they're typically the different groups are identified by the structure that holds the spores. And here are some examples, none of which you have to know because it's in black ink as opposed to red ink. Just know that there are different many different groups, and typically identified by the structure that holds the spores. And here you see some examples of that. So let's move on. Sexual reproduction has its own unique characteristics in fungi, and we're not going to get into the details of that. Here's one example. This is two different filaments coming together, two different hyphae from a male and not male and female, but plus and minus, and that is the spore structure right in the middle here. This is that was a zygospore. This is an ascospore, the basidiospore. There are all kinds of spore types. Now, in terms of disease, which is why we study this, fungal diseases are called mycoses. Singular is mycosis and plural are mycoses. Typically, these are chronic infections. There are 5 different groups of fungal infections or mycoses, and they are classified based on the tissue involved and the mode of entry. It is often much more difficult to treat bacterial fungal infections than bacterial infections because of the fact that fungi are eukaryotic organisms, whereas bacteria are prokaryotic organisms. Prokaryotic organisms have characteristics that are quite different from eukaryotic, and those can be targeted by certain therapeutic drugs, which will not have any effect on the eukaryotic host, the human, because they only target prokaryotic mechanisms. It is more difficult to treat mycoses because you're trying to target a eukaryotic type of organism inside another eukaryotic organism. So many types of treatments will have some side effects on the host cells. The 5 different types of mycoses are as follows, and I told you it is determined by location. Systemic mycoses are throughout the body of the infected person. They're deep within the tissues. Subcutaneous mycoses are just beneath the skin. Cutaneous mycoses affect keratinized structures that are associated with the skin, like the hair, the top layer of the skin, the nails, and are typically accompanied by inflammation. There is a subtle but important difference between cutaneous mycoses and superficial mycoses. The superficial mycoses are also localized to keratinized material associated with the surface. However, in this case, there is no or very little tissue reaction. So the person that has the superficial mycoses doesn't react hardly at all. Whereas with the cutaneous mucoses, there will be inflammation. And then finally, the superficial mycoses, which are, and then finally the opportunistic mycoses, which are caused by fungi that are normally non-pathogenic and but will cause disease in individuals who are immunosuppressed. So there we have it: systemic everywhere, subcutaneous below the skin, cutaneous associated with the skin and causing inflammation, superficial associated with skin structures associated with skin, again, but no tissue, very little tissue reaction, and then opportunistic mycoses in immunosuppressed individuals. You would think that immunosuppressed individuals could almost have any kind of fungal disease, but they typically have certain types of fungal diseases associated with that immunosuppressed state. There are medically important phyla. These are the phyla. Again, based on the structure of the spore-containing organisms, they're divided up into these different groups. Some can have members that cause disease, so they're medically important. And these are examples which we won't get into. Another type of somewhat different life cycle. Another example, again, subtle differences in the life cycle. And another, so they were actually zygospores, ascospores, and basidiospores produced by these different main groups. But again, you don't need to know that, and you can see how there are similarities. This is the group that would form this kind of mushroom, and again, the mushroom is the sexual reproductive structure producing spores and is a tiny, tiny part of the organism. You see it as the main structure, but most of the organism is below ground in a mycelium. Now, let's get into certain groups that have characteristic organisms that cause important disease processes in humans. Anamorphs include these kind of fungi that only produce asexual spores, but that's not the important point here. The important point are the examples. Coccidioides is a fungal disease which is a fungal organism that causes coccidioidomycosis. There's a mouthful for you. It is a systemic, serious mycosis. It spreads throughout the body, slowly progressing, and will eventually kill the person the treatment is long and complicated. It is sometimes called San Joaquin Valley disease because the spores are found in dust, particularly in the San Joaquin Valley in California. Pneumocystis is the genus of an organism that also causes a systemic mycosis. It is the kind of thing that you typically find in someone also who's somewhat immunosuppressed. And then Candida albicans causes a cutaneous mycosis, candidiasis, which is a very common occurring fungal infection and easily treated. Okay, that's it for fungi, organisms that we'll look at later in more detail. The algae, there are no algae that cause an ongoing disease process. There are some that are toxic and can indirectly poison people. Algae are eukaryotic. Again, we're covering eukaryotic organisms, are eukaryotes. Unicellular or filamentous or multicellular. They can be really huge even, or just single cells. Most are photoautotrophic. They're light fixing in terms of energy. They use light for their energy, and they use carbon dioxide mostly for carbon source to build up organic molecules. We're just comparing in this table fungi and algae. But let's move on. All kinds of algae grow in the ocean, of course. There are all kinds of sizes. The thing is that some cause disease, as you'll soon see. There are organism structures called diatoms. They're unicellular, and they produce a toxin called domoic acid. These single-celled organisms that cause problems in humans, typically what happens is that you'll get them in the local water, and they get taken up by filter feeders, mussels, invertebrates, shellfish, oysters, whatever, clams. And those organisms that humans eat will concentrate the toxin and can cause problems. Another example are with dinoflagellates, again a unicellular algal organism. It has a neurotoxin which can cause a paralytic shellfish poisoning, not paralyzing the shellfish or poisoning the shellfish, but the shellfish filter it out of the water as a food source, and when humans eat the shellfish, they get poisoned because it concentrates in those shellfish. And so we leave that, and we've covered fungi and algae, and we move on to protozoa. As you know, protozoa are also called protists. Although the still very popular name is protozoa. They are single-celled eukaryotic organisms that are neither plant nor animal. They are chemoheterotrophs. So eukaryotic, unicellular, chemoheterotrophs, an extremely diverse group of single-celled organisms. Often they will have a vegetative form, which is always called the trophozoite. They will reproduce by both sexual and asexual reproduction. And some, not all, but some will form cysts, which are very hard coatings that allow them to survive in adverse conditions like dry, in a dry state. There are many medically important groupings, phyla of protozoa. There's a tremendously huge variation in all of these, and you'll see when in the examples that I give you of medically important organisms that are protozoal single-cell organisms. All right, now you don't have to know the name of the family. You do need to know with all of these, you do need to know, including the previous algae, fungi. I think we had 3 in the fungi and 2 in the algae. You do need to know the organism's name. This is Giardia lamblia. This is giardiasis, a gastrointestinal infection. And Trichomona vaginalis, which causes trichomoniasis, a genitourinary infection. Many of these infections are identified by under a microscope from samples taken from the patient, sometimes by identifying the vegetative form or sometimes the cyst form. So Giardia causing giardiasis and Trichomonas causing trichomoniasis, GI and GU. This, I'm not going to get into this organism. This is an organism which is an amoeba. It is Entamoeba histolytica, which causes intestinal amoebiasis. I have to tell you that there are gigantic numbers, like for Giardia, or trichomoniasis, or amoebiasis, intestinal amoebiasis, huge numbers of infected people throughout the world for all of these diseases that I'm going to mention. Much more than you ever imagined. And we'll get into numbers when we go into the disease processes later in the course. So, Entamoeba histolytica causing intestinal amoebiasis. Plasmodium vivax. There are several different Plasmodium organisms, not just vivax. So there's several different species members in this genus, the genus Plasmodium, that cause the disease malaria. This is one of the more common ones. Cryptosporidium, an opportunistic infection that often will occur in immunosuppressed individuals, people who have poorly controlled acquired immunodeficiency syndrome. Let's move on. Each of these organisms, each of these protozoal organisms has, or protist organisms, has a unique life cycle and often complicated. This is the example of Plasmodium, which causes malaria. It has a insect vector, meaning it gets carried from person to person by an insect that has the organism in it. The organism lives inside the mosquito's salivary glands, and when a mosquito bites you, it injects a little bit of its saliva into you, and if it's infected with Plasmodium, it will also get the organism into you. It injects a little bit of saliva because its saliva has a anticoagulant present in the saliva, so that the blood doesn't clot and prevent the mosquito from sucking its blood meal. So the organism gets into you and travels to the liver, and then where it multiplies, it then goes into red cells. And another innocent mosquito comes along and takes a blood meal and gets infected, and then flies, and then the organism grows in its salivary glands and then flies off days later and infects somebody, bites somebody, infects somebody else. The disease is identified by structures inside the red cells, these ring-like structures inside red blood cells. Red cells, of course, do not have a nucleus, so it is very apparent if there are red cells that are infected in the person. They'll have this structure inside them, and it shows up very easily. And here again, the whole process is quite complicated. You don't need to know the details other than the key things I told you about the mosquito vector and the red cell and ring structures. Cryptosporidium, not going to get into it. We're not going to get into this one either. Trypanosoma causes trypanosomiasis, also called sleeping sickness. It also has an insect vector, the tsetse fly, and it's throughout Central Africa. There are huge areas that they can't. It'll also infect cattle. There are huge areas that where it's a big problem. Leishmaniasis, another protozoal infection caused by Leishmania. Again, a huge problem. Not going to talk about that. Okay, so we've covered the fungi, algae, and protozoal organisms, and now we'll talk about helminths or worms. Worms are parasites, and we study them in microbiology. I took an entire course also on parasites. Again, life cycle can be quite complex and somewhat different in each organism, certainly, and easily confused. Not an easy topic, but there are certain fundamental characteristics about these worms that you should know, and some of the diseases associated with them, the ones that cause the disease, and etc. So that they are helminths or worms, eukaryotic multicellular animals. They are chemoheterotrophic. Essentially, they are parasitic worms. There are two important groups in this. There are two important phyla in this group of animals. One are the platyhelminthes, platy meaning flat, so flatworms. They're called flatworms. That includes flukes, which cause many diseases, different types of fluke organisms, and tapeworms, which also several different types that cause disease in humans, and the nematodes, which are roundworms. So you have platyhelminthes or flatworms and nematodes or roundworms. Typical key characteristics of parasitic worms are that they, because they're parasites and they live inside their host animal, the host animal provides quite a few things that directly to the animal, so that the animal doesn't need certain structures or abilities that most animals do have. Many lack a digestive system. After all, if they're living in the gastrointestinal tract, they don't need for the food to be digested. The host animal digests, or human digests the food for them, and they can just absorb it through their surface structures. They have a reduced nervous system because they are very well protected and don't need it. They don't need to see, they don't need to hear, they don't need to do much except for move and reproduce. And even then, often they don't move much. Sometimes they just attach to the wall. So they have a reduced locomotion system or locomotory system. They all typically have a very complex reproductive system that is highly productive. Many eggs or offspring are produced. And I can tell you that the majority of the world's population is in fact parasitized with worm infections. Most people in the world have worm infections at any given moment. So we'll talk about the platyhelminthes, and we mentioned that there were one important group were flukes, and here is an example, and we'll move on. And here is a life cycle of one particular type, and we won't get into all the details other than the fact that there are often one or more intermediate hosts, and then the definitive host. So in this case, the definitive host is a human, and the intermediate hosts, there are a couple of intermediate hosts. One is a snail, and the other is a crayfish that eats the snails. and the human, the definitive host, eats the crayfish. The definitive host is where the sexual phase will occur, and they release eggs in the GI tract, and it gets into the water, so it gets in the snails. Many fluke organisms, you don't need to be a fisherman fishing on the ocean to get this kind of fluke. Some fluke organisms live in ditches and infect snails, and then the organism goes from the snail and burrows through the skin into the human. Okay, the other major group of flatworms were not flukes but tapeworms. So we had flukes and we had tapeworms. This is the head of a tapeworm. A tapeworm is an interesting animal. It has segments, and each segment has its own sexually reproductive organs in it. And each segment has both male and female structures, and so it can produce eggs efficiently and in giant numbers, and those eggs then go into the feces in the GI tract. They're excreted out in the feces and picked up by another organism that gets that eats the egg. So it's typically a oral fecal route of contamination from one individual to the next. No eyes, no feet or legs, doesn't have to have a sophisticated nervous system or digestive system or locomotory system, but has a very sophisticated reproductive system. And here's an example. In this case, this type of tapeworm, there is a definitive host, which is canid or dog, wolf, etc. And there are two possible intermediate hosts, but we're not going to get into this. Now, the other major group, those were platyhelminthes, flatworms, which could be flukes or tapeworms. And the other major group of helminths are nematodes, also called roundworms. A very commonly occurring nematode infection in humans are pinworms, which are approximately half a centimeter to a centimeter long, depending if you're looking at the male or the female. The female is typically larger than the male and has extensive genital reproductive structures. Another example is Trichinella spiralis, which causes the disease trichinosis, and it infects pigs and can easily be picked up by humans. The organism gets into the human's GI tract, then grows for a while, then burrows into the tissue and forms a cyst in the tissue. It does the same thing in the pig, and that's how humans pick it up in poorly cooked infected pork. Okay, let's just pause for a minute. Now, we have been talking about we talked about fungal, algal, protozoal, and worm or helminth, parasitic type of diseases. And some of them that I mentioned, particularly the protozoal infections, involved an insect vector, and those insects are called arthropods. They act as a vector to transfer the disease. We talked about Plasmodium vivax carried in female mosquitoes that take a blood meal. They pick up the disease and it goes to the salivary glands and reproduces, and then they'll infect another individual when they bite the other individual and inject some of their saliva in. This lovely thing down here is a tick, which can carry tick-borne diseases. There are a number of different protozoal diseases carried by ticks. Mites also carry disease. There's lice and fleas. Some have 6 legs, some have 8 legs, but these are all arthropods, and they're acting as vectors. So these are vector-transmitted diseases. Here you see a louse. That's the singular for plural lice, and there's a flea, fly. Some of these can carry disease. This is another type of insect